Hello, my dear friends and NEET aspirants. A very warm welcome to our amazing Power Hour sessions. I am Dr. Ami Bhayani, your mentor and guide for the zoology sessions of Power Hour. Now, what are these Power Hour sessions? These are power-packed sessions where we are going to be discussing the most important and core concepts of zoology. These are those concepts where either students tend to make little mistake or some important points of the NCERT which are skipped while we are reading them, or these are the ones where we find it a little difficult to understand. But worry not, this is what we are here for, and let's get started with one of those important topics of zoology. So let's get on board. Now, during all the sessions of zoology, we will be starting today with biomolecules. However, we will be covering almost all the important concepts of every chapter of zoology. Today, as I said, we are starting and opening up a very new perspective and an interesting perspective of the chapter called biomolecules. Now, this is a chapter which even our chemistry faculties and colleagues will be discussing with you all, as it is the last chapter of your 12th standard in chemistry. However, 9th chapter of zoology in NCERT standard 11. Is this chapter important from NEET point of view? Absolutely yes. Why? Because as we can see from the statistical data of the PYQs, you can see that almost 3 to 4 questions are asked every year. So it's a high weightage chapter from NEET point of view. Today, we will be discussing 3 important concepts. One, the chemical analysis part, which is in the introduction part of the chapter, the proteins, and then the enzymes. Why these 3 parts? Because these are the 3 ones wherein we are having large number of questions coming in the NEET paper. Okay. So let's understand what are initially biomolecules. Now, when we talk about biomolecules, we know bios means life. The molecules which are organic compounds, the carbon containing organic compounds that are obtained from the living tissues is called as biomolecules. Okay. Now, the first question arises that as you studied in botany, there are large number of diverse group of living organisms that we see around. Are all the living organisms made up of the same elements? The answer would be yes. When we do a chemical analysis, we found out that all the living organisms are made up of similar elements, similar compounds. The next question comes, is the Earth's crust also having the same elements? To our surprise, we will be surprised to know that the Earth's crust and the living organisms on elemental analysis showed that are made up of same elements. However, obviously, yes, the composition and the percentage will vary. So what is the difference? Let's have a look at it. Now, when we take a table wherein we have the percentage weight of certain elements in the Earth's crust and in the human body, you will see that hydrogen, carbon, oxygen, nitrogen, and sulfur are more abundant in human body as compared to in the Earth's crust, whereas elements such as sodium, calcium, magnesium, silicon are more abundant in the Earth's crust as compared to the human body. But if you pay careful attention to the table, you will find that when we talk about oxygen, see, in the Earth's crust, the most abundant is oxygen. Similarly, in the human body also, the most abundant is oxygen. So this can come as our MCQ, which you need to remember. Also, we need to remember that nitrogen is very little in the Earth's crust as compared to human body, and silicon negligible amount in the human body as compared to Earth's crust. We do not need to remember all the values because they've never asked in the MCQs in NEET. But the most abundant and the least abundant is important to remember. Done? So let's proceed. Now here we are going to talk about our human body and the living organism's body. So now if we are talking about the living organism's body, we know everybody is made up of cells. Every living organism has the simplest basic unit of life, that is cell. What are the components of the cell? So if we see the components and their percentage within the cell, we can see that water is the most abundant chemical in the cell, forming 70 to 90% of the total cell. Proteins, the next most abundant organic matter, having 10 to 15%, nucleic acids 5 to 7%, carbohydrates 3, lipids 2%, and ions 1%. This is an entire table given in the NCERT as table 9.4. Now, what do we need to remember here and pay attention is that when we talk about the most abundant, be careful, chemical, and most abundant biomolecule. In the paper, though we are stressed, but pay careful attention to what are they asking. Most abundant chemical, if they are asking, our answer should be water. 70 to 90%. If they are asking most abundant biomolecule, the organic compound present in the cell is our proteins. So most abundant chemical, water. Most abundant biomolecule, proteins. Is that clear? Moving further ahead. When we talk about our body, is our body made up of organic only inorganic, or both? As we saw previously, in the table where we discussed between the Earth's crust and the human body, table 9.1, we saw that there are organic as well as inorganic compounds. So his query, whether there are organic, inorganic, yes, there are both organic as well as inorganic components in the body. But here in this chapter, we will be discussing only about the organic compounds. Okay. Now, how do we know what are the organic components present in our body? So let us do a small but an interesting study and a small experiment. What are we going to do is we are going to take a small mortar and pestle. A mortar and a pestle. We are going to take a small piece of a vegetable or liver, because we know that every living organism has the same components. Now, we will take this mortar pestle, take a small piece of liver tissue or a vegetable tissue, add trichloroacetic acid, Cl3CCOOH. Now, when we add trichloroacetic acid and we grind it in mortar and pestle, we will obtain a thick slurry. What are we going to do is, we are going to take a cheesecloth or a cotton cloth and we are going to strain the slurry. When we strain the slurry, as we know that whenever we strain anything, some things will get filtered out and some things will remain in the cloth. Similarly, here also, something will get filtered out, something remaining in the cloth. What gets filtered out means, yes, it is acid soluble pool. Whatever is dissolved in the acid will get filtered out. Whatever is not dissolved will remain in the cloth. So, whatever is filtered out is acid soluble, or also called as filtrate. Whatever is not dissolving in the acid is the acid simple insoluble pool and is called as the retentate. Now, what will dissolve and what will not dissolve? Let us have a look at it. Now, when we see what dissolves and what does not dissolve, anything that has small molecular weight will be dissolved in the acid and will get filtered out. Whatever are large molecular weight substances will not get filtered out. But small and large is very subjective. So, anything which has a molecular weight of less than 1000 daltons are basically the ones which we call it as micromolecules, or simply biomolecules, as says the NCERT. Whereas anything which has a greater molecular weight, more than 1000 daltons, will remain in the acid soluble, insoluble pool. However, everything has an exception. So why not this? Here in the acid insoluble pool, We said everything that are large molecular weight substances, or I can call them as macromolecules, will remain, but what will also remain with them is lipids. Now, what is the exception with lipids? We'll talk about it in some time. Let us first have a look further ahead with acid soluble and insoluble. Since acid soluble we discussed is micromolecules, they are also monomeric substances, that is, which cannot be further broken down. We all know that when monomers join together, they form polymers. So these polymeric substances will come in the acid insoluble pool and non-polymeric or monomeric in the acid soluble pool. So in the acid soluble pool, we have monomers of proteins, that is amino acids, of carbohydrates, sugar, of DNA, RNA or nucleic acid, nucleoside and nucleotides. Also, pay careful attention that inorganic components like sulfates, phosphates, etc. are also found in the acid soluble pool. In the acid insoluble pool, we have polymers of, that is proteins, polysaccharides, nucleic acid, that is DNA and RNA, and as I told you, lipids. So together, when we talk about soluble and acid insoluble pool, it constitutes the entire composition of the cell. Now lipids, what is it that is different about lipids? Now lipids basically have a molecular weight of less than 800 Daltons. Then why are they in the acid insoluble pool? See, when we talk about lipids, they are found in the cytoplasm. They form the cell membrane as well as the organelles membrane, as you've studied in the cell chapter. When we grind it, what happens is it forms small spherical structures, which we call them as vesicles. Now, have you ever taken a glass full of water, try putting little oil into it and stir it? What will happen? Oil will form small circular structures. Similar circular structures called vesicles will be formed when we grind it with trichloroacetic acid. Now these vesicles, being water insoluble, cannot be filtered out and remains as retentate. However, we have to remember what is given in the NCERT, that lipids are strictly not macromolecules. Still in the acid insoluble pool because it forms vesicles and is water insoluble. Now this can come as our assertion and reason, so make a note of it in your NCERTs. Okay? Moving ahead, since we've discussed about the basics of what is present in the body, organic, inorganic, let us have a look at the question. Now, the question is assertion and reason. Assertion saying, living organisms have more of nitrogen and oxygen per unit mass than the Earth's crust. And the reason being, living organisms have more of calcium, magnesium, sodium in them as compared to the Earth's crust. I'm giving you a time of 15 seconds because it's very easy. So let's solve it. Try solving it in 15 minutes and send me the answers. Okay, I'm waiting for your answers. Okay, so let's see. Amino acids, they are monomers. Yes, they will be found in the acid soluble pool. Polysaccharides, polymers. Nucleosides, yes. Phosphates, we studied inorganic, yes. Nucleotides, yes. Monosaccharides, yes. Lipids, no. They are micromolecules, but not in the acid soluble pool. Lipids, no. RNA polymers, no. Sodium, yes. So there will be 1, 2, 3, 4, 5, and 6. So answer would be 6. Is that clear? Yeah. One more question. Let's go for it for the next 15 seconds. You have matched the column where in one side you have 4 options in column 1 and column 2 has 2. Try matching them. आ रही है आवाज बराबर? कंटिन्यू करो, वेट करो. किसी का भी वही देखो. मेरे टाइम पे ये सब होना था. Okay, so the number of R group, when the R group varies, it could be any R group, because it is this R group that changes, rest remains constant in an amino acid. When R group changes, I will have different amino acid, so I can replace R with anything, any kind of a group. So then I will have multiple types of amino acids. Type of amino acids depends upon what? It depends upon the R group. So, will I need multiple amino acids in my body to form proteins? No, I don't need multiple. So, how many I need? I need 20 amino acids to form proteins in my body, and these amino acids that I need in my body to form proteins are called proteinaceous amino acids, or I call them as standard amino acids. Is that clear? So, R group could be any, number of amino acids multiple, in the human body 20, known as proteinaceous or standard amino acids. Now, these proteinaceous amino acid word is given on page 106 on the first line in NCERT, which some of the students tend to miss. So, be very careful with it, mark it in your NCERT. Moving ahead, we can also classify amino acids on the basis of number of amino and carboxyl group. How do we classify them? If the number of acidic groups is more, carboxyl group is more as compared to amino groups, you call it as an acidic amino acid. If the number of basic groups or NH2 groups are more, you call it as basic. If the amino and the carboxyl group number is equal, you call it as neutral amino acids. There will be innumerable examples which will come here. However, we stick to the ones which are given in the NCERT. Acidic amino acid example is glutamic acid, basic lysine, neutral is valine and glycine. We can also replace the R with an aromatic group, and we have examples such as phenylalanine, tyrosine and tryptophan. Now this can also come as our MCQ. There is one more way of classifying amino acid is on the basis of synthesis in our body. Now on the basis of synthesis, we classify it into essential and non-essential amino acids. Now essential, as the name suggests, are very, very important. Not that these are not important, but comparatively these are more. Why? Because our body cannot synthesize it on its own, and we receive these amino acids from our dietary source. The proteins that we eat are the dietary sources of these amino acids. However, the non-essential amino acids are the ones which our body can synthesize. So hence called as non-essential amino acids. So one we studied is the basis, what depends upon the type of amino acid, or what decides the type of amino acid is the R group. Like any other compound, there will be physical and chemical properties of amino acids. What decides the physical and the chemical property? Is it only the R group? No. Be careful. This again can come as our MCQ in the NEET, where they will tell us that it depends upon dash. So it depends upon R group, amino group and carboxyl group. Is that clear? Moving ahead. Multiple properties are there, but one of the most important property of an amino acid is that it behaves as a zwitterion. Let us understand what is zwitterion. Now we said amino group has an H, NH2, COH and a variable R group attached to the carbon. Now when we place this amino acid in an acidic pH, what will happen? The NH2 is a very good hydrogen acceptor. It accepts the hydrogen and behaves, as you can see, as a positively charged ion. Similarly, when we put it in a basic medium, the COH releases its H plus and behaves as a negatively charged ion. At a particular pH, there will be an intramolecular hydrogen transfer where COH releases its H plus and that is taken up by the NH2. So at this pH, the amino acid has a positive as well as a negative charge and is now behaving as a dipolar ion, or we also call it as the Zwitter ion. Is that clear? Now why it behaves as a Zwitter ion is because of the ionizable nature of the NH2 and the COH groups, and hence in solution of different pH the structure of amino acid changes. Now here if you see we have the word hence. This can come as our assertion and reason. Remember, wherever in the NCERT you come across statements with the word as, because, hence, thus, since. These can come as your assertion reason. Now that is all about the understanding of amino acids. They are monomers. They will join together to form a polymer. How will they join? Let us have a look at it. Now we have this as one amino acid, the second, the third, the fourth. Whenever proteins are formed, proteins are polymers of amino acids. Polymers can be homopolymer or a heteropolymer. Homo where we have the same monomer units repeating. Heteropolymer is where different types of monomers are joined together to form a polymer. Always remember, protein is always a heteropolymer and never a homopolymer. It is always a heteropolymer. So we have different amino acids which are joining together. How do they join? The carboxyl groups OH of one amino acid and the H of the NH2 of the second amino acid form a bond, and there will be a water molecule formed which will be released. This process is condensation, leading to formation of a bond here, and this bond is called as peptide bond. Two amino acids joining together, dipeptide, three tripeptide, and multiple ones, polypeptide. So here, if we write an N, that is multiple amino acids, then what it forms is a polypeptide. Now if we look at the polypeptide chain, the first amino acid has the amino group which is free and is not participating in the bond formation. The last amino acid, carboxyl group, is free, not participating in the bond formation. So the first amino acid is called as the N-terminal. The last amino acid is called as the C-terminal. This was a question last year NEET 2023 paper where they said that the first amino acid is C-terminal, last amino acid is N-terminal. It was a false statement. First amino acid N-terminal in the polypeptide chain, last amino acid C-terminal. Now, how do we decide and how many types of proteins are there? Now you must have seen in the newspaper such kind of games that come where you have to form words. Now in these alphabets, if you form words, say if you have TAP, tap, if you make it the other way, it is PAT, pat, TEAT, EAT, eat, TAR, tar, RAT, rat. So if you are just changing the alphabet, the word changes. Similarly, if you change the sequence of amino acid, the protein changes. So if I just reverse the sequence, as you can see in the first two, the type of protein changes. So what decides the type of protein is the sequence of amino acid. Type of amino acid, R group, type of protein is the sequence of amino acid. Again, a very important question for our NEET. Be very careful. Proteins, this is how proteins are formed. When we talk about the structure of protein, see, our chemistry friends will describe the structure and study structure in different ways, be it organic, inorganic, or physical. We as biologists will study it at 4 levels. The primary structure, the secondary structure, the tertiary structure, and the quaternary structure. Let us study each one of them. Now we know that proteins are formed within the cells with the help of an organelle called ribosomes, which are protein factories. Now when proteins are formed, as I told you, they are heteropolymers. They will form a rod-like structure like this, where you have different monomers, different amino acids joining together to form a chain of amino acids. This is the primary structure of the protein, occurring as a rigid rod-like structure. However, it does not remain as a rigid rod, but it folds upon itself to form what is called as the secondary structure of protein. There are 2 types of secondary structures of proteins, called as the alpha helix structure and the beta plated structure. Now when we talk about the alpha helix structure, as you can see in the diagram here and in the model here, we have a chain of polypeptides where there is an intramolecular hydrogen bond. That is hydrogen bond between amino acids of the same molecule. So there is a single chain, intramolecular hydrogen bond, forming a helical structure, which is like a revolving staircase. And there are only right-handed helices present. So remember, this can again come as our MCQ, that there are only right-handed helices present, forming the alpha helix structure. Then what is a beta plated structure? Where we have, as you can see here, 2 polypeptide chains. 2 polypeptide chains. These 2 polypeptide chains will have intermolecular hydrogen bond. Intermolecular hydrogen bond forming this kind of a structure, which is called as the beta plated structure. These basically form what is the structural protein in our body. Moving ahead to the tertiary structure. When the long polypeptide chain folds upon itself to form a 3-dimensional structure, as you can see here and also here, when it forms a 3-dimensional structure, say like this, it gives a hollow woolen ball-like appearance. This kind of a structure is called as the tertiary structure of protein. Okay. Now proteins don't just exist as primary, secondary, tertiary. Tertiary structures will also come together. More than 1 polypeptide chains in the form of tertiary structures will be arranged in the form of a circular structure, in the form of a cube or a plate, to form what is called as the quaternary structure of protein. Examples given in the NCERT is hemoglobin, where we have 2 alpha and 2 beta chains, is also insulin, which we will study in biotechnology, where we have chain A, chain B. In proinsulin joined together by chain C. In insulin, chain C is removed. We will discuss it in detail in biotechnology. Now, this is about the structure of protein, which can come as match the columns. Moving ahead to which are the most abundant proteins. Now, most abundant protein could be in the animal world or could be in the biosphere. When we are talking about it in the biosphere, the most abundant protein in the biosphere is rubisco. Can you tell the full form of rubisco? Yes, ribulose, bisphosphate, carboxylase, oxygenase. You will study about it in botany. Whereas, if we talk about the most abundant protein in the animal world in our body is collagen. Collagen, again in structural organization, we know it forms the intercellular matrix. Okay. Now, let us understand about the functions of proteins. When we are talking about the functions of proteins, proteins have innumerable functions in our body. Collagen, which is the most abundant one, forms the intercellular matrix. Trypsin acts as an enzyme. Insulin is a hormone. Antibody, the defense system in the body, fight against infections. receptors which will help take up different sensations and receptions, also the place where hormone binds, present on the cell membrane. GLUT4, this is again a PYQ. Please remember, GLUT4 is the one that will help in glucose transport within the cell, is also an important function. Now, when we are talking about the functions of proteins, one of the most important function of protein is that it behaves as enzymes. It acts as enzymes. So let us now cover up another subtopic from where a large number of questions are asked, is enzymes. But the question is, are all enzymes made up of proteins? Till now we thought that yes, all enzymes are made up of proteins. However, now we know that it is almost all enzymes made up of proteins, not all. Again, true and false statement, you have to be careful. Then what are the other types, or what are the other components that will work as enzymes? There are certain nucleic acids which will work as enzymes. Example is ribozymes. But in this chapter, we are not going to talk about ribozymes. My dear children, we are going to be discussing about proteinaceous enzymes. Okay? So let's understand the enzymes. Before that, let us also have one question before we move into understanding of enzymes. Okay? So it's a match the column, where in column 1 we have most abundant proteins in biosphere in animals, type of proteins, type of amino acids. On the other side, we have the other examples. Let's take a time of 30 seconds and quickly solve this. Okay. Let's see. Chalo. Most abundant protein in the biosphere, if you're talking about that, the answer is rubisco. Most abundant protein in the animal is collagen. Type of protein, if you're talking about what decides the type of protein, is the sequence of amino acid, that is 3. And what decides the type of amino acid is the R group, that is 4. So the answer will be 2, 1, 3 and 4. Let's see which option has that. So 2, 1, 3 and 4, the answer is C. Yes, so most of you all have answered C. Very good. Yeah. Now when you're talking about enzymes, as we said, enzymes are basically tertiary structure proteins. What are enzymes? Enzymes are basically tertiary structure proteins. Now these enzymes which are tertiary structure proteins means they will have a woolen ball-like appearance. When they have a woolen ball-like appearance, as you can see here also, there are lot of pockets, lot of crevices which are there. So here also if we see, here we have one crevice, here is another, here is the third, here is the fourth one. Okay. However, only one of them, as you see here, only one of them is the place where that substance will go and bind, which has to be converted into product. Now that substance which has to be converted into the product is called as a substrate. So the enzyme at the active site, the substrate binds. Active site is that place or that pocket where the substrate binds, and it will be converted into product. This shape of the active site is decided by the bonds formed between amino acids. So it will be highly specific in different enzymes, and it will be extremely specific for the substrate that binds and also for the reaction that it catalyzes. It acts as a biological catalyst. So it speedens up the rate of reaction. Now, how much fast does it speeden up? Is it little here and there, or is it too much? It is to such an extent that no reactions can function without enzyme in the body. Let us take an example. If we are talking about the fastest acting enzyme, the example is carbonic anhydrase. Now, this we will study even when we study the respiration chapter, that is breathing and exchange of gases. Carbon dioxide, when combines with water, it will get converted into H2CO3 in the RBCs and to a small extent in the plasma. Now, if there is no enzyme, that is, in the absence of enzyme, 200 molecules will be converted into H2CO3 in 1 hour. However, in the presence of carbonic anhydrase, it is mind-boggling to see that 600 molecules are converted in 1 second. 600 molecules in 1 second. It is unimaginable. It increases the speed by 10 million times. Again, this can come as our MCQ. How do we say the rate of physical or chemical reaction? So basically it is the chemical reactions that are catalyzed. When we talk about the chemical reactions catalyzed, the rate can be depicted as delta P by delta T, which is the amount of product formed per unit time. That is 6 lakh molecules per second. That is the amount of product formed in per unit time. Okay. It is that fast that it speeds up the rate of reaction. The question is, how does it do this? So let us see. To have it converting into the product, the substrate into the product, now the enzyme has a particular active site. We denote enzyme as E, we denote substrate as S, we denote product as P. At the active site, the substrate will diffuse and go towards the enzyme. So you have to remember that the process is diffusion. When the substrate diffuses towards the enzyme, it goes towards the active site. Once it reaches the active site, there will be slight change in the shape of the active site and it will bind fitly or tightly to the active site. Once it binds tightly to the active site, it forms a complex which is called as ES complex. Now this is a transient complex, that is, it is not going to remain as it is, but it is an obligatory formation that has to take place. Once the ES complex is formed, the enzyme will now either make or break the bond as per the requirement and convert it into an enzyme product complex. Once the enzyme product complex is formed, the product will be released and the enzyme is now ready to bind with another substrate and convert it into the product and repeat the cycle. This works like a lock and key mechanism because, as I told you, the shape of the active site is specific for the product that or the substrate that will bind to it. But when we say that enzyme substrate complex is formed, converting it into an enzyme product complex, what is exactly happening that it speeds up the rate of reaction? Now, any substrate which is there, when it has to be converted into a product, the substrate has to be provided a particular amount of energy. That energy, we call it as activation energy. Once you provide a particular amount of energy, the substrate will reach to a high energy state, which is called as transition state. Once it reaches the transition state, only then it will be converted into the product. So every reaction, be it a making reaction or be it a breaking reaction, it has to pass through the transition state structure. Now, so there is a particular amount of energy required. Now what is enzyme doing? Enzyme is reducing this energy barrier. Say for example, I give you a huge rock. I tell you throw it and convert it into small pieces. Standing on the ground is not possible to do the same. So you take it on the 20th or the 25th floor and from there you drop it, you obtain your product. But you have to take it to the 25th floor, means provide a particular amount of energy. Now if our engineering friends are making a particular machinery where on the 5th floor, if we take the same rock, but from the 5th floor, with the help of that machinery, it is throwing it with a particular force, velocity at a particular angle, it will convert it into the product, but with the utilization of less amount of energy. Is that possible? Yes. This is though a physical process. Same analogy will be used to understand what enzymes do. So in the enzyme, the substrate will be converted into the product, but the amount of activation energy is reduced. So the energy barrier has been reduced. Is that clear? Thereby converting the substrate to product fast and more easily. So do we need to do this in exothermic as well as endothermic reactions? Yes. Now if we look at the same graph where progress of reaction on one side and the potential energy on the other side, if you see the substrate is here, and in the graph A, product is at a lower potential energy level as compared to the substrate. What is happening means substrate, when it is getting converted into the product, energy is lost. When energy is lost, this kind of a reaction is called as exothermic reaction. In the NCERT, we also have the term mentioned, which is calling it as the spontaneous reaction. Spontaneous reaction. Similarly, if you look at the graph 2, in the graph 2, you can see that is graph B. Substrate is present here at a particular energy level. However, product is at a higher energy level. So substrate converting to product is gaining energy. So you are providing energy, and hence you call it as an endothermic reaction or energy requiring reaction. Is that clear? So on the basis of that, let us now quickly solve a question. Read the following statements and arrange them in the correct sequence. Substrate binds to the active site of the enzyme, fitting into the site, followed by binding of the substrate. You have to choose which is the correct sequence. Second is binding of the substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. Active site of the enzyme, now in close proximity of the substrate, breaks the chemical bond and the substrate, and the new enzyme product complex is formed. Enzyme releases the product of the reaction, and free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. Giving you 15 seconds, solve it and let me know the answer. We have Amruta who has answered. Yes, Harshali has answered. Very good. Sanvi, very good. Sabha, very good. Okay, let's see. Yeah. So substrate binds to the active site of the enzyme, fitting into the active site. Yes. Followed by what will happen now? There will be change in the active site shape. Again, correct. Followed by active site of the enzyme, now in close proximity, breaks the bond. And then you have that the product is released and the enzyme is again free. So the order is A, B, C, and D. And our first option itself has that answer. So that will be our correct understanding and correct answer for the question. Moving ahead to understanding of the last part of the topic, which is factors affecting enzyme activity. Will there be any factors which will affect the enzyme activity? Yes. There are factors such as temperature, pH, concentration of substrate, or presence of inhibitors. How do they alter and why do they alter? Let us see. Now, when we talk about temperature, be it any enzyme, every enzyme has a specific temperature at which its activity will be to the maximum. That temperature is called as optimum temperature. Now when we plot a graph with temperature on one axis and enzyme activity on another axis, we obtain a graph which is a bell-shaped graph. Meaning any activity which is on either side, that is above or below the optimum temperature, we can see that the enzyme activity reduces. Why does this happen? When we increase the temperature, say more than 40 degrees Celsius, what is happening is the weak bonds which form the tertiary structure of proteins are broken down, converting the tertiary into secondary, secondary into primary, and thereby changing the structure or altering the structure of the protein chain, and thereby the enzyme activity reduces, or there is no enzyme activity above 40 degrees Celsius when we are talking about our human body, because the proteins are getting denatured. So the enzyme activity stops above 40 degrees Celsius. If we are reducing the temperature, again the enzyme activity reduces and the enzyme becomes temporarily inactive. Now, with all the organisms, living organisms, is it 40 degrees and above that the enzyme activity will stop? There are exceptions again, wherein there are some organisms such as Thermus aquaticus bacteria which live in extremely high conditions such as in hot vents and sulfur springs. Now these organisms, since they are living in extremely high temperature, the enzymes also work at a temperature of about 80 to 90 degrees Celsius. They do not get denatured, and the enzyme which we are going to study again in biotechnology is Taq polymerase enzyme. In PCR we will be talking about them where we need them. So when we talk about organic biocatalysts, these are basically thermolabile because they are highly sensitive to temperature. You call them as thermolabile. Except we have Thermus aquaticus bacteria's Taq polymerase enzyme. But if you are talking about inorganic catalysts, they have a capacity to work even at high temperature, and hence we call them as thermostable because their structure is not going to get damaged with the temperature or higher temperature. Similarly, when we are talking about the pH, there is again an optimum pH where we will have maximum activity of the enzyme. Any pH above or below the optimum will, as you can see, reduce the rate of reduce the enzyme activity, giving a graph which is a bell-shaped graph. Now the third is substrate concentration. Now, what do you think? If you keep on increasing the concentration of the substrate, will the enzyme activity increase? Again, if we take a graph where on one side we have the substrate concentration, on the other axis we have the velocity of reaction, as we keep on increasing the substrate concentration, the rate of reaction keeps on increasing till it reaches a particular value which is called as Vmax or maximum velocity, post which the enzyme activity does not increase. Why so is because, again because means it can come as an assertion reason, the number of enzyme molecules are less as compared to substrate. So as we keep on increasing the substrate concentration, it goes and binds to the active site of the enzyme. Once all the active sites of the enzymes are saturated, there are no free active sites where it will go and bind. Thereby, once it reaches the maximum velocity, that is once it has reached the stage where all the active sites are already saturated with enzyme, there will be no further increase in the velocity. And hence this kind of a graph. In the NCERT, in this graph, if you see, there is something called as Km that is mentioned. Now what is this Km? Km is Michaelis-Menten constant. It is that particular concentration of the substrate at which we attain half of the maximum velocity. That substrate concentration is called as Michaelis-Menten constant. Moving ahead to the last part of the topic where we are going to talk about the presence of inhibitors that will affect the enzyme activity. Now inhibitors, as the name suggests, are chemicals or substances in the presence of which the enzyme activity will be affected, it will be reduced or even stopped. Now competitive means it is competing. Competing with what? Competing with the substrate. There are certain chemicals whose structure is almost or very closely resembles the substrate structure. So they will compete with the active site of the enzyme. They will go and bind with the active site of the enzyme, not allowing the substrate to bind. So obviously, if the substrate does not bind, the enzyme activity reduces. And that is the reason they will inhibit the enzyme activity called as inhibitor, because they are competing with the substrate. Since they are closely resembling the structure, you call them as competitive inhibitors. But is this a reversible or an irreversible reaction? It is a reversible reaction. When you increase the substrate concentration in the presence of an inhibitor also, it will displace the inhibitor and again come and bind with the enzyme. Thereby, we will be able to have the maximum effect of the enzyme and the maximum enzyme activity. So, we will be able to reach Vmax. However, we will have to increase the substrate concentration. So these are the factors which are going to affect the enzyme activity. Now there is an example of the competitive inhibitor given in the NCERT, where there is an enzyme called succinate dehydrogenase. This will convert succinate into fumarate. You've studied it in botany. Now succinate is a substrate, succinic dehydrogenase is the enzyme, and this is the product. However, closely resembling succinate is another molecule which is malonate, which is an inhibitor. In the presence of malonate, succinic dehydrogenase will have its active site wherein malonate will come and bind and succinate cannot bind. So the amount of fumarate formed will reduce. Again, this can be reversed as we have discussed. This is very important because twice it has come in the NEET paper 1 last year. So it's a very, very, very important PYQ which we need to understand. Another example given in the NCERT is competitive inhibitors are used to control bacterial pathogens. Again, this can come as an MCQ, a very small line at the last part of the paragraph. So be very careful and read it and underline it. So that's all from my side for today. But in the end, let us solve one more question and then have the closure for this. So assertion: high temperature decreases the enzyme activity. Reason: proteins are denatured at high temperature. High temperature decreases the enzyme activity. Is it true? Yes, it will decrease and then stop the enzyme activity. Why? Because they are denatured at high temperature. This is an NCERT statement with the word because. So if there is, as I told you, because, hence, thus, as, and since, it is a correct explanation. So assertion is true, reason is true, and reason is the correct explanation for the assertion. So I hope today's session was helpful for you, was helpful in the sense that you were able to now understand the concept well and will be able to answer in your final NEET exams. So with that saying, let's meet in the next session and bye for now. Thank you.